Hello, everyone. My name is Natasha Brinkley, and I'm a bioengineering major working under the mentorship of Dr. Jin Nam. Today, I'll be presenting my research on implementing polymer microwells for optimizing neurite outgrowth and engineered functional nerve tissue in vitro. For some context, there's more than 600 neurological diseases that afflict humans, whether congenital, geriatric, or traumatic in origin. Many of these pathologies are rare or somewhat poorly understood, so researchers have mostly relied on computational and animal-based models to characterize and develop therapies for the conditions. But both of these approaches have major drawbacks. Computational models, like the one shown above, are inefficient and can never accurately capture the complexity of a living system. And animal models, such as rats, have inherent physiological differences from humans that can make translating results unreliable. But recently, researchers have developed a new strategy: neural models that are grown in vitro from human cells. These models have demonstrated promising morphological and functional similarity to native tissue, and they have the crucial advantage of patient specificity. Since the nerve tissue is grown from human neural stem cells, or HNSCs, the cells can theoretically be extracted from any patient to facilitate the design of a custom diagnosis and treatment plan. The premise of this project is based on a few significant findings from prior studies. The first is that culturing HNSCs on static PVDF-TRFE fibrous mats promotes differentiation into neurons. PVDF-TRFE is a synthetic polymer that's biocompatible and piezoelectric, which means that it accumulates electrical charge in response to mechanical stress. The scaffolds are fabricated through this really cool technique called electrospinning, which basically involves ejecting liquid polymer into an electric field and onto a collecting platform. The polymer gets pulled into long threads in the process and built up as a mat of fibers, sort of like how cotton candy is made. Our electrospinning setup also has a collector that spins at several thousand RPM, so when the fibers are ejected, they get deposited onto the wheel in aligned parallel rows, just like the SEM figure you can see on the right. The second major finding is that mechanoelectrical stimulation of HNSCs can actually induce multiphenotypic differentiation. Healthy nerve tissue is made of three different types of cells: neurons, oligodendrocytes, and astrocytes, all of which are pictured on the right. Oligodendrocytes and astrocytes mostly serve to support neuron functionality and are typically grouped together as something called glial cells. When HNSCs were cultured on a piezoelectric scaffold and treated with hydroacoustic actuation, they simultaneously differentiated into all three cell phenotypes. But not only did the cells just differentiate, they also self-assembled into a working neuroglial interface that mimicked native tissue. Instead of just relying on transient surface charges to do the work, like the previous static study did, the researchers here applied enough mechanical force to generate a biologically relevant electrical potential. But even though the resulting nerve tissue was highly functional, it still exhibited poor alignment and stunted axonal growth. So, given the limitations I just mentioned, the objective of this project is to establish a reliable protocol for optimizing the alignment, length, and connectivity of neurite outgrowth in engineered nerve tissue. To accomplish this, I designed a cell culture system that integrates PVDF-TRFE electrospun scaffolds, PDMS microwells for cell colony control, mechanoelectrical stimulation, and a regimen of biochemical growth factors and inhibitors. Part one of this experiment was to optimize the application regimen of nerve growth factor and ROCK inhibitor that would be administered to the cells. NGF is an axon agonist, or promoter. It serves to accelerate axon outgrowth by negatively regulating the activity of the growth cone actomyosin. Axon extension is moderated by restraint mechanisms, so when we weaken that restraint a little bit using NGF, we can substantially enhance growth rates, as seen in the figure next to it. When nerves get injured, the body responds by stimulating this massive proliferation of glial cells to form neural scar tissue. During the process, neuronal axon growth is suppressed, so we use a glial antagonist to create the opposite effect. We suppress glial proliferation to enhance axon outgrowth. It's also important to note that the behavior of ROCK inhibitor is highly dose-dependent, and like all signaling molecules, it performs a variety of self-regulatory and contradictory roles. We perform the optimization itself through a Taguchi multifactorial and multi-level experimental design, represented by these tables on the left. Over a 2-week culture, 4 NGF and ROCK inhibitor ratios were applied at 3 different levels and staggered at different application times to make 9 unique conditions with 2 samples per condition. Those graphs display the relative values of axon length and myelination between each condition. We found that the optimal treatment regimen for axon length was A2, B3, C2, and D3, And for percent myelination, it was A1, B2, C1, and D1. But unfortunately, these results can only be considered preliminary data. Using this sort of experimental design, we need to repeat the study to confirm the validity of our conclusion. Part 2 of this experiment was to design a PDMS microwell mold for guided cell seeding, and to then perform a long-term cell culture study to assess the effect of that seeding on axon growth and alignment. When I first started working with HNSCs, I noticed that the longest and most well-aligned axons tended to form either as bridges connecting adjacent colonies together or as outgrowth from the colony edges. So when we were brainstorming ideas to optimize the axon formation of our nerve tissue, we proposed the idea of seeding the cells in artificial colonies. Our hope was that the cells would sort of naturally be inclined toward bridging between those colonies in neat little lateral sequences. That way, we could control the direction that they travel in and incrementally increase that traveling distance to effectively maximize the elongation of their axons. The simplest execution of this concept would be to make a sort of cell stencil. It would have little perforated microwells that cells could be deposited into to force them to stay secluded in colonies. To fabricate that microwell stencil, I designed several iterations of a 3D printable mold in SolidWorks. Once printed, the cast could be filled with a liquid polymer that takes on the shape of the perforation as it cures, and can then be peeled off as a thin, pliable substrate. My earliest iterations, which seem a little bit ridiculous in hindsight, had individual impressions for each microwell, with little rails bracketing either side to secure it on top of the scaffold. You can see this in the first and second molds along the bottom. It was impossible to peel the polymer out of these molds once it had cured because the details were so tiny and just the edges were too small. So most of my later versions involved just a flat plane with intermittent protrusions sticking out of it. And this just meant that the microwells would need to be cut out with a scalpel before being placed on the scaffolds, but it's okay. A little bit more work for more accurate results. To print the mold, I used a 3D DLP resin printer, specifically the Anycubic Photon Ultra model, rather than a traditional extrusion printer. Resin printers tend to have a higher maximum resolution. Ours is on the order of around 20 microns, and the well dimensions of my mold are around 400 by 1200 microns, so I needed the features to be as well defined as possible. For the microwell material itself, we chose PDMS, or polydimethylsiloxane. PDMS is a silicon polymer that is biocompatible and non-cytotoxic, which means that it won't kill the cells that it comes in contact with. It's also highly flexible, easy to work with, rapidly cures, and it has tunable mechanical properties. Before pouring the PDMS, we first coat the mold with photoresist coating to prevent it from sticking. Then we can peel the cured PDMS off as just one big sheet with those little holes punched in it. The electrospun scaffolds are only 5 by 10 millimeters though, so we need to cut the microwells out to fit using a scalpel. In our very first long-term culture study for 5 weeks, we used gap distances of 500, 1,000, and 1,500 microns. Fortunately, the experiment was successful, otherwise I wouldn't be here today. We had healthy secluded colonies, cell proliferation and differentiation, and visible axon bridging between those colonies. This proved that our project was indeed feasible, so we could move on with optimization trials later. The only minor issues that we needed to address were that the cells proliferated pretty far beyond the borders of their colonies, and the axons still weren't quite long enough. The fluorescent stain sample shown actually had an initial gap distance of 1,500 microns, but the cells spread out so much over the culture duration that it became more like 800 microns eventually. But after tons of troubleshooting, my most recent 5-week study yielded some pretty impressive results. The green stain shown on the right is GAL-C, which indicates lipid-rich regions, in our case, myelination. The red stain is beta-tubulin 1, also known as 2J1, which marks neuronal bodies and axons. And the composite staining stacks the two images together to indicate regions where both are present. In other words, it lets us visualize where the myelin wraps around the axons of our neurons. So just from these images, we can clearly see consistent alignment, myelinated axons, which indicates the presence of mature differentiated neurons and glial cells. We can see strong bridging between the colonies and just this crazy long axon outgrowth. Our longest observed neurite outgrowth was about 1,900 microns, just basically 2 millimeters, which is very impressive. Just for comparison, the study that this project was building off of was only able to achieve a maximum neurite length of less than 500 microns. So it's 4-fold larger. Even though I'm presenting the completed track, this research is really only the tip of the iceberg. My next step is to translate all of these optimized parameters to a 3D cylindrical conduit culture for modeling functional nerve morphogenesis. Up until now, I've demonstrated that we can grow multiphenotypic nerve tissue with long connective axons and strong alignment in a 2D culture system. But real nerves aren't two-dimensional, so my next major goal is to make this model as physically realistic as possible. The conduit will be formed by taking one of our PVDF TRFE mats and rolling them into a sealed tube, sort of like making sushi. The conduit will be filled with a conductive hydrogel that can sustain mechanoelectrical stimulation and is firm enough to localize cell colonies in suspension. That optimized gap distance that we found earlier with roughly 2 millimeters will be used to inform the implantation intervals of our HNSCs, so they'll be injected into the tube at 2 millimeter increments from each other. Our optimized biochemical factor regimen will be applied to enhance axon growth, and eventually a concentration gradient will be established along the length of the conduit to hopefully guide the direction of nerve morphogenesis. And that's it for some brief acknowledgements. Thank you so much to my PI, Dr. Nam, my graduate student mentor, Yui Tai, my research colleagues at the Treat Lab, and to everyone involved with the Undergraduate Research Engagement Committee for giving me the opportunity to conduct and share my work with this community. Thank you.